Good evening, everyone. I'm Andy Bolan. I'm the Associate Director for Patient and Medical Community Engagement at ProQR Therapeutics. In this talk, it'll be about 5 minutes. I'm going to take you through what we, as a small biotech based out of the Dutch city of Leiden and Cambridge, Massachusetts, are doing in RNA science. And Maria's given a wonderful overview about that. Usher syndrome specifically, and other retinal diseases. As a pharmaceutical company, we have to have forward-looking statements, and here they are. So who is ProQR, really? We're dedicated to developing RNA therapeutics for patients suffering with severe genetic retinal disorders with the mission, should I say, of stopping vision loss or even trying to reverse some of the symptoms. In the field of RNA therapeutics, ProQR is at the forefront at trying to deliver these targeted therapies. Our RNA therapies are antisense oligonucleotides, and Maria kindly covered that as well, and I'll come back to it later, which are specifically designed to correct the underlying cause of a disease in a person's RNA. Now, RNA stands for ribonucleic acid, and it's an essential component of all living cells. RNA is used for translation, in the process of which a proteins are created in a cell. RNA is produced from DNA. I always find it harder to describe RNA, and I always have this struggle with my mum when I'm trying to describe it, because everyone kind of understands what DNA is, having watched Jurassic Park, and knows what you do if you cross a toad with a mosquito millions of years in the making. So RNA really takes what the DNA is, translates it into proteins to create healthy cells. You know, ProQR was founded in 2012 by our current CEO, Daniel de Boer. A few years earlier, Daniel and his family were faced with a diagnosis of cystic fibrosis for his newborn son, an incurable rare genetic disease. Daniel sought help from experts in the field of drug development and formed ProQR to help people like his son. When viable treatments for cystic fibrosis came through for Daniel's son, he looked at other areas of clear high unmet need. And since 2017, we've seen ProQR concentrate and bring forward clinical research, i.e. research involving individuals living with Leber congenital amaurosis type 10 or LCA10, Usher syndrome type 2A, and autosomal dominant retinitis pigmentosa. As of January this year, we fully enrolled our pivotal phase 3, and Maria talked about the different phases. So the pivotal, the last stage, in LCA10, which sets the company into a new era where we could have data that we can take to the likes of regulators such as the FDA, EMA in Europe, and MHRA here in the UK to register a therapy for people living with LCA10. A very exciting time for the company. And I have some more exciting news later about Usher syndrome type 2A. We know that it is estimated that there are over 2 million people living with inherited retinal diseases, and there are very few treatments. We think there might be more because of a lack of genetic testing, accessibility to genetic testing. And we believe the best way as a company to fulfill this mission is to develop RNA therapies for people with rare genetic eye diseases. We've built a platform to develop these targeted therapies to address the underlying cause of the disease and try to improve vision. For the vast majority of these 2 million people, we know that there is no treatment available for vision loss and eventual genetic blindness. With more than 300 genes identified so far, and Maria described the difficulty in there are other genes we haven't found so far, of inherited retinal diseases, there is an immense opportunity for us to develop therapies for an area of clear high unmet need. So how do we do it? We have a unique platform in the field of RNA. Maria's touched on it earlier. ProQR is trying to develop medicines that we can use intravitreal delivery. Our RNA therapies use antisense oligonucleotides, which are specifically designed to correct the underlying cause of a person's RNA, to stop the disease progression or even reverse symptoms. What are antisense oligonucleotides? They consist of short stretches of synthetic RNA that are chemically modified to increase efficiency and uptake into the cell. This technique is an established approach to treat genetic diseases, and there are RNA-approved therapies being used to treat patients around the world today. Why is it different to the kind of gene subretinal injections? Well, our IV injection, for a drug to work, you have to put it into the right part of the body. our RNA therapies work best when they're delivered directly to the affected organ, which in this case is the eye. In the case of retinal disease, RNA therapies can be injected into the vitreous, which is the jelly-like substance in the cavity filled with the jelly-like fluid. This delivery method is known as intravitreal injection. IV injection is one of the most common procedures for eye diseases. The procedure is performed by eye doctors for common conditions at the moment, such as age-related macular degeneration, you might have heard of, diabetic retinopathy, and retinal vein occlusion. IV is different from subretinal injection, as Maria kind of outlined, which requires surgery. But before I close, as I had to get through that pretty quickly, I wanted to update you on the news from today, which is exciting. I'm pleased to announce positive results from the trial that Maria showed earlier, the phase 1/2 in Usher syndrome and non-syndromic retinitis pigmentosa, the clinical study STELLA, which has met all its stated objectives. Now, I didn't deliberately ask the team to announce the great news today, knowing that I was coming on here, but it certainly has helped in making me have such a good mood while presenting. This is a crucial milestone in our ongoing inherited retinal disease research for ProQR. And the reason is we have one in LCA10. This is a second inherited retinal disease that we've now shown that our platform works, which is wonderful for us as a company. There are currently no treatments for more than 16,000 individuals with vision loss due to Usher syndrome type 2A and non-syndromic RP due to the Usher type 2A exon 13 mutation. And Maria talked about that. The STELLA study is a phase 1/2, so early, and we were looking at safety. Clinical trial for the catchily named QR-421A molecule. We definitely need to come up with a name now that we've seen that it slightly works. To test it was safe and leads to better vision. The interim findings that we did last March actually, were encouraging. A year on, the positive results have come out today. With only one injection of QR-421A, the STELLA study showed benefit in the treated eye compared to the untreated eye across all participants who took part. We've seen this benefit across all participants in the STELLA study, which included clinical trial participants with advanced vision loss, as Maria described earlier, as well as those with early and moderate vision loss. Now, we judged that by best corrected visual acuity, or BCVA, in the severe advanced population. And for the early to moderate, we had retinal sensitivity, or OCT, as Maria gave earlier, which also showed positive results. Having shown these to the FDA in the US in the last few days, we've agreed with the medical regulators to advance QR-421A to the final stages of clinical testing. This will be our second clinical program to move to the final stage following our LCA10 program. The news today really validates our RNA platform, and everyone at ProQR is extremely pleased with that. Based on that, we'll be conducting 2 final stage clinical trials, phase 2/3 trials, and we'll work with regulators in the coming weeks and months to submit clinical trial protocols for the 2 final stage registration trials called Sirius and Celeste. No doubt you've picked up on the galactic theme, Stella, Sirius, and Celeste. Something I always put down to the very sci-fi loving ProQurians we have, as we call ourselves, or I could use a cheesy line like we're searching for the stars in our research. I will let you decide. The reason we're having 2 final studies in phase 3 is to test the advanced populations in Sirius and the early populations of Usher syndrome type 2A in Celeste. Both will be counted as registrational trials, which is really great news. The FDA has already accepted that. I have whistled through this. I have a specific briefing, which I've actually shared with Chloe already today, as well as other retinal organizations around the world, which will take you through in a plain language the results from today. So if any families or anyone has any interest in wanting to learn more about what we announced today, that document is available. But with that, I'd like to close my presentation. I'd like to say thank you to Chloe and the Usher Kids UK team for inviting us along to showcase, ProQR as a company that's trying to bring the wonderful research that's going on in inherited retinal diseases to life and into people. And yeah, a big thank you to following Maria, who described the science really well. So thank you.
Thank you, Andy. It was brilliant to hear, and great timing to be able to hear those results. Really excited. I guess one question for our community is, in the Celeste trial, you mentioned patients who have less advanced disease progression. What age would they be eligible to participate in that?
Yeah, so I mean, like, the logistics of all the trials is going to be figured out in the next coming months. What I can say is our Stella trial was 18 and over. However, we have just kicked off a pediatric study in LTA 10. So we, as a company, I know, having listened to Daniel talk earlier today at the results, you know, there is an ambition to have a pediatric element to it, but it's very early to talk about the kind of protocol or what it's going to look like in the future.